Right. Thanks, Andrea, for the introduction. So first off, I'm recovering from a cold, so my voice is doing funny things sometimes, so please bear with me. Alright, so GMOs, it's a subject that a lot of people have heard some things about, some negative things sometimes, but a lot of people don't really know what they are, and so that's the goal of just today, to kind of walk you through what are GMOs and why are they being made. And so first I want to put it into some context. And so start with a crop that everyone knows really well, so corn or maize. So this is entirely man-made. So this is not a natural plant, and it probably wouldn't even survive in nature if we were no longer there. And this was not made in a lab, it was made 6,000 years ago by Mexican farmers using a plant called teosinte, so which is a kind of a weed. It's very small. It makes these small seeds encased in a hard shell. And so they improved it into a better version that we still grow today, which is maize. So if you want to learn more about this, my colleagues outside have a booth near the popcorn machine where they can tell you all about this, and you can get some popcorn while you're there. So this is true for many other crops. So tomato used to be really, really tiny, like berry-sized. So again, over thousands of years, early farmers turned it into much bigger crops that are easier to grow. And so I kind of wanted to point this out, so because there's a long history of modifying plants and crops to make them better. And before I delve into what GMOs are, I kind of want to quickly explain how we improve plants through breeding, which is what we still do as well today. Just with a simple case, so say that you have so we have a plant, a maize plant that we grow out in the Midwest. It's very high yielding, very optimized. But then a disease comes in, which you can see here on the sleeve, that has symptoms. It can be a virus, a bacteria, an insect. So then we need to make this plant resistant to the disease. So typically what was done is you would look for a maize variety from somewhere else that is naturally resistant. We have seed banks, we can find these. And then you would cross these two varieties. The resulting plant would be resistant to the disease, but it will also carry, so just like when two people have a child, it will carry both genomes. It will have both the good and the bad from both parents. And so that's also what's here. So it's typically going to be much smaller. So you have to keep crossing it back over generations and generations to the plant that you want to make it look like that again. So this is a process that can take easily 10 years. So traditional breeding works really well, but it's slow. So in the 80s, scientists figured out GM, so genetic modification, so which is essentially a way, so if you would want to achieve the same thing using GM, what we would do is we would study this plant that is resistant to the disease. We would figure out why is it resistant. And this is often one gene that tells the immune system of the plant, this is the virus that you need to be looking for, kill it when you see it. And so what we can do is we can isolate this one gene, literally in a test tube, and we can introduce it into the plant that we want to modify. And so the result is a plant that is in every way identical to the plant we started with, except it will have one single gene added to it that will make it resistant to the disease. And this takes about a year. So it's much, much faster. It's very precise. So now, the part that people worry about is transgenics. So this is where you do the same thing, but you take the gene not from another maize plant, but you can take the gene from any organism. And so, so a case of this, which is very, very widely grown all over the world. So there's this bacterium called BT. So the bacterium is very widely used in organic agriculture. It kills caterpillars. It's very, very toxic to caterpillars, but completely harmless for us. So what you can do is you can take the one single gene that is responsible for killing the caterpillar, put it into maize. The result is a maize plant that will produce its own natural biological insecticides. So you no longer have to spray with insecticides. Which, so in this case, is a very good thing. But so this is something that people worry about, because it is possible that you can get unintended consequences. because you're taking a gene from one organism and you're putting it in another organism. And so it is possible that you could get unintended effects. This is why these plants are regulated very heavily, and they're tested very heavily for all kinds of potentially harmful effects. So all of these transgenics are tested by the USDA, the FDA, the EPA, before they are released. So one really good case for GM is what if there is no natural resistance, which sometimes happens. So a very good example of this is papaya in Hawaii. So papaya is very sensitive to something called ring spot virus, and that nearly wiped out the papaya industry in the early 90s. There was no natural resistance, so the only solution to this was to use GM. So to introduce a little bit of the virus DNA into the plants, in the plant's genome. So to tell it, if you see this DNA, kill the virus, because it means that you're infected. And this saved the papaya industry. Something similar is now going on with oranges in Florida. So citrus greening is now wiping out orange orchards. No natural resistance, so people are now also looking into a GM way to solve this. So in some cases, GM is really the only way of dealing with problems in agriculture. Because this is a question that you sometimes hear, why do we need to modify plants? Why do we need to mess with plants? So one thing is diseases, so we need a new flu shot every year. So plants need to keep their resistance up to date. We just need to produce more food, population grows. And just the way we produce food relies very heavily on a lot of water, fertilizer, pesticides. So if we can make plants that require less insecticides, less water, that would be great. So a very clear example of how damaging agriculture can be is, so what you see on the left, it's the Aral Sea. It used to be a lake the size of Lake Michigan out in the Soviet Union. Today, 50 years later, it's gone. All the water has been used up for irrigation. And so this is a huge catastrophe in the region. So if we can make plants that require less water, we can avoid these types of situations. And so then I just very quickly want to end with what we do. So I'm in Dave Jackson's lab. So one of the things we study is essentially how many seeds can you pack on one column? And can we improve that? And so we work with these mutants that are very ugly, so they tend to be very short, very disorganized. Obviously, you could never use this in agriculture, but so you can see they make many rows of seeds. So what we can do is we can figure out what is going wrong here, and then dial it down up to the point where you're still making corn with a few more rows of seeds, but it's otherwise it looks completely normal. But add this up over an entire field, and this is a huge increase in the number of seeds that you produce. So this is the kind of knowledge that we generate that can then be used to improve plants, either through breeding or through GM approach. And that can help us make more productive crops. Okay, so this was a very fast kind of introduction into GM. So there's lots of stuff I couldn't cover, so if you have any questions, I'll be more than happy to take them. Yes?
Yeah, I'm not sure I understand this. So if there's a disease that threatens a plant,
yeah.
you take a weakened pathogen and put it into the...
no, that's the way we do it in vaccines. So the way we do it in plants is usually you're going to try to make a resistant variety. So we put a gene in the plant that kind of activates the immune system of the plant. So it will recognize the virus and tell the immune system to attack it.
Okay, and how do you find these genes?
So either they're, so maize grows all over the world, so some of it is naturally resistant. Like if you have a lot of this virus in a certain part of the world, the maize there will be resistant, so we can take a gene from that. Or we can use like bacteria, like the case I showed before, which have these genes in them that are insecticidal.
Have I heard you're not supposed to feed your children genetically modified food?
So all the genetically modified food that is now out there has been tested very rigorously, as I said. So it has been deemed safe by the US government. It has been tested very, very securely. So everything that is now on the market is safe. Because it went through an enormous amount of tests to make sure that it's not allergenic, that it's not harmful to the environment, not harmful to anyone who would eat it.
What about the issues with monoculture? Are you nervous about that?
So monoculture is, so it's exactly this. It's the way we grow crops today. In the Midwest is these huge areas of the same type of plant, essentially. Because everyone buys their seed from the same company, to a certain extent. And this makes it very, so you can get diseases like this that really ravage through the whole field. Yeah, this is an issue. It's in some ways an issue separate from GMOs because this is just the way we grow plants nowadays, and you can use GM for monoculture as it has been used, or for more small-scale culture. In a way, it's a separate issue from GM. There's no more questions. Oh yeah, please.
How do they check for possible human effects? You said these are well tested. How long do they test them for? Do they go through generations or a month or two or what?
So they usually use animal models to test first. So you can go through, you can do long-term studies with mice because they don't live very long normally. So that's usually the first type of test that they do. So they will use animal models in the same way that we test drugs, which we also do on animals that are kind of close to us. They also just do a lot of lab tests to make sure that it's safe.
Well, they recall the drugs frequently because they find that their testing is imperfect.
Yeah, it doesn't happen that often. For GM, there's no known case of anything slipping through that shouldn't have. But yeah, there is always a potential risk. So it's weighing the risk of how much can we improve the way we do agriculture versus the acceptable risk of GMOs that we have. But it's impossible to exclude any risk. But everything that we have today, as far as we know now, it's perfectly safe. And a lot of this, we've been growing for 20 years. So we would know at this point.
If you were to speculate, how far do you think this GM could go as far as application of plant?
Excuse me, I didn't catch that.
How far can GM go as far as applying to plants, for instance? So, just speculate with whatever you think are the limits of the possibility.
They're virtually endless. It's pretty much what we can come up with. What I'd like to see is to have a GM applied to make plants more nutritious, have them produce, for instance, vitamins, which we can totally do. So those, but you can do pretty much anything that you want. Because we know how to do all this stuff in the lab. It's just a matter of getting it out and getting it tested.
So any specifics related to that he's worked on?
Yes, there's, so for instance, in having vitamin-producing plants, so there's this thing called golden rice, so which is rice that produces lots of vitamin A, which is very important in Southeast Asia, because a lot of children have vitamin A deficiencies, and they rely very much on rice. So if you can make rice that produces vitamin A, you could get vitamin A into their diets in a really easy way. So that's something that is currently it's out there. So those types of applications are made because we know a lot about how plants work and how biology works, so we have all the understanding we need to improve the plants, I think, at this point.
Does a specific gene have only a single function, or
That's a very good question. It depends on the genes. So most of the genes that we use are very well studied. So for instance, the insecticide I showed, so this is the gene is a blueprint for a protein, and the protein is toxic to caterpillars. As far as we know from all the tests, that's the only thing it does. But like I said, it is possible to have unintended consequences, which is why you should test these things.
is there any way of testing something? How could you determine under what circumstances it might have a difference?
It's fairly empirical. So you put the gene into another organism, and then we test everything we can test about that organism. And usually it behaves perfectly the way we expect it to. So in a way, we've gotten quite good at predicting these types of things. But yeah, it's a very good question, and it's obviously a concern. All right, thanks everyone. I'll be around if you have any more questions.